Hi, everyone. My name is Vikas Gupta. I'm the business leader for the Gene Therapy Initiative here at Repligen. Welcome to the 2020 Repligen Virtual Seminar series here, focusing on gene therapy. And many of you must have attended our face-to-face roadshow over the years held globally. And to all the attendees, either the first-time ones or the attendees that are coming back, welcome again. So this particular seminar will be focusing on some of the latest trends that are occurring in the very dynamic field of gene therapy. And we'll be talking about some of the primary drivers that are causing the changes, some of the early adopters of new technologies that are impacting the gene therapy workflows, as well as how we as partners can assist towards the progression towards gene therapy manufacturing 2.0. So for those of you who might be looking at Repligen for the very first time, so Repligen as a company has been around since the 1980s, and it was primarily focusing on the production of proteinuria ligands at the time. And we made a conscious effort to pivot towards being a bioprocessing solutions provider from 2012 onwards. Our focus always has been to differentiate ourselves by offering disruptive technologies and products that offer a unique value proposition to our customers. You will be hearing about a lot of these products and technologies during the course of the presentations today, including alternating tangential flow, TFTF, and slope spectroscopy-based concentration measurements technologies that we have in our portfolio and are being used in the field of gene therapy manufacturing. So if you look at the entire gene therapy workflow on the upstream side, where there is a focus on upstream process intensification, our products like ATF and TFTF are being used on the downstream side, both for TFF steps, including our systems, as well as our filtration modules, both hollow fiber and flat sheet cassettes-based products. And on the analytics, our slope spectroscopy is being used for plasmid concentration measurements, as well as viral vector concentration measurements. And most of our product portfolios are being offered in a single-use format that facilitate gray space of bioprocessing. They have fully scalable platforms to enable your bench scale process development to full-scale clinical and commercial manufacturing. And the products are being extensively used for all kind of gene therapy technology platforms, they be based on AAV or lenti viruses or mRNAs, as well as for some CAR T applications as well. And if you look at the gene therapy market as a whole, I'm sure you guys are very well aware of some of the more recent gene therapy and CAR T therapies that have been approved over the years, including Kymriah that was approved back in 2017, Yescarta from Gilead, Luxturna, which was part of Spark and now part of Roche, and the latest approval that happened in 2019 for Zolgensma for spinal muscular dystrophy, from Novartis. And when we look at these 4 recent approvals, actually the FDA's outlook on the gene therapy market is extremely positive. They are planning to hire almost 50 new clinical reviewers to fast track some of these approval processes. And they are expecting, by the year 2025, almost 10 to 20 new approvals per year. And even in terms of the INDs, they were expecting almost 200 INDs every year from this year onwards. And a quick look at the gene therapy pipeline here, a very, very robust pipeline. This includes cell therapies that use viral vectors. So we have almost 1,000+ trials going on in the preclinical trial phase. And for phase 1, phase 2, and phase 3, again, a very robust pipeline. And the phase 1 number is almost actually equal or on par with the number of trials going on for the mAb space. So a lot happening here, and many of these gene therapy processes are actually scaling up to that 500 to 2,000 liter scale, which is the sweet spot for all of the single-use manufacturing technologies that are being used in the bioprocessing world today. Another important thing to note, if you look at the graphic on the right-hand side, is that typically we have been used to seeing the mAb manufacturing timelines extending to 8 to 10 years, or for certain orphan drugs, from 6 to 8 years. But with gene therapy, especially with this advanced therapy status, the RMAT designation, these timelines are getting squeezed to 3 to 5 years. So there is a very rapid therapy discovery and development process, and the technology adoption is happening at a much faster pace than ever before. All right, so looking at the agenda, first and foremost, I wanted to thank all the speakers who have given their time to walk us through their talks today. And we'll be starting with the talk, which will talk about the use of TFDF technology to improve the yields using lentivirus vector production as an example. And then the subsequent talks, the two subsequent talks, will be talking about TFF applications, the use of KR2I systems to help with some scale down studies, and the selection of the appropriate filtration technologies when it comes to hollow fibers, as well as flat sheets, which one is the best format for AAV versus lentiviruses. And then we'll wrap up the seminar today with an introduction to the flow VPE technology, in this case for bacterial growth measurements. But the technology, as I said before, is being used very extensively for plasmid concentration measurements, as well as for viral vector concentration measurements. So now I'll hand over the baton to Renee Gantier, who is the Director of Technology for Gene Therapy here at Repligen. Thank you very much and enjoy the seminar.
Thank you, Vikas, and good day, everyone. I'm René Gantier, Director of Technology for Gene Therapy at Repligen. Thank you for joining us today. We have a great agenda and great speakers for this event to illustrate and demonstrate how technology-driven process intensification and optimization will bring us to the gene therapy manufacturing 2.0. For the next few hours, our speakers will be presenting their sessions. These conversations were pre-recorded with the speakers so that they could be present in the attendee chat alongside other participants. The attendee chat is the place to ask questions and share thoughts. The speakers and Repligen representatives will address as many of your questions as possible during the sessions, and we'll follow up post-event with any questions that we cannot get to. With this format of event, you can choose to experience it as interactively as you want. You're welcome to focus on watching the sessions, focus on writing into the chat, or combine the two in any way you find comfortable. Please remember to be courteous and professional with your thoughts and questions. So it's now my pleasure to introduce our first speaker, my colleague, Michael Bransby from Repligen. Michael is the Research and Development Director of Process Technologies, and today he will be discussing a novel approach to intensify the viral vector production from suspension cell culture using the tangential flow depth filtration technology.
Greetings, and thank you for joining me for our Gene Therapy 2.0 technical seminar. My name is Mike Bransby, and I'm the R&D Director for Filtration, working primarily out of Repligen's Southern California office in Los Angeles. Our topic today is centered around increasing bioreactor productivity of lentiviral vectors for gene therapy applications, which was done in partnership with Oxford Biomedica, using a very new filtration technology called TFDF. This is just one application of this technology, and we don't have a lot of time together today, so I'm going to focus on the yield benefit and then describe in general terms that the technology is fast, requires a very small footprint, and is operational simple even at larger scales. If you'd like to learn more about how the technology works or other applications, I encourage you to contact your local Repligen sales representative or visit repligen.com. The title of the webinar is Increasing Lentiviral Vector Bioreactor Yield with Simplified Execution Using Tangential Flow Depth Filtration. As we survey the viral vector workflow, Repligen has developed and launched many solutions to help with different pain points. If you're looking for a faster or better way to complete your unit operation or analytics, give us a call. Today, we are focused on viral vector production and specifically the clarification steps, where there are some clear needs in the industry. I've been in the gene therapy area for over 15 years, and the harvest step has always been a great challenge. Can we get increased viral vector yield with gentle methods that are lentivirus compatible in a closed aseptic format? Hopefully, you'll see that we're addressing several of these needs with the new TFDF technology. Okay, the case for an increase in viral vector yield is supported by a model built and published by Renninger and others in December of last year. Their model incorporates the number of active clinical trials, average yields, and requirements per indication to create a plot of the number of batches required per year. The projected demand for viral vector is plotted in orange, while the supply, how much can be produced worldwide, is in blue. Two different models were created with different rates of capacity growth. But both models, however, conclude that total demand will exceed total capacity even this year. So if you look, you know, the curve in the orange is going up almost exponentially. That is, the demand is great, but the capacity is flattening off. And so there's this big disparity. So you've heard about flattening the curve for COVID, which we all want to do, and it keeps going up and down. We want to keep the COVID curve flat, but we don't want to keep this curve flat. We want to get capacity supply to meet demand. We believe TFDF is a potential way to do that for your processing. Oxford Biomedica was the first to develop a commercial lentiviral-based gene therapy delivery system, and they have transitioned their adherent HEC-293 cells to suspension bioreactor in order to increase yield. Repligen is a leader in high-impact bioprocessing technologies, the latest of which is TFDF, or tangential flow depth filtration. TFDF is the first depth filter that runs in tangential mode. In order to accomplish this, the filter is tube-shaped and is not a dead-end filter. In this picture, we have a bioreactor that contains blue HEC-293 cells and orange lentivirus particles. The bioreactor feed is connected to the bottom of the tubular TFDF filter. Permeate, shown here in orange, travels through the wall of the tube, while retentate continues through the lumen and the end of the tube, and then is directed back to the bioreactor, not shown here. So you have the blue cells recirculating back into the reactor and the orange lentivirus particles coming out. An integrated system with pumps and software runs this automated process. This brings us to the two key questions raised when Repligen's TFDF technology met Oxford Biomedica's suspension culture. First, can TFDF improve the harvest unit operation over classic depth filtration? And second, because the cells are preserved, that is, they're continuing in the reactor, they're not getting stuck in the filter, can they be used to create more virus in a high productivity mode, which is also frequently referred to as batch perfusion? You can read about the detail of the work in the Cell and Gene Therapy Insight article published this year. The first experiment is a straightforward comparison of TFDF and depth filtration at the 5-liter scale using a therapeutic lentivirus. When normalized against bioreactor titer, depth filtration delivered a yield of approximately 70%. They're in the salmon color. TFDF in comparison delivered a yield greater than 90%. Clearly, the TFDF transmits the virus. But why the increase in the yield? Well, there are several reasons. The first is that the nominal pore size of the TFDF filter is 2 to 5 micron, meaning that while holding back the cells, the 100 or 120 nanometer size lentivirus particle has plenty of room to navigate the channel. Second, that the filter material, which is completely synthetic, primarily built of polypropylene, does not bind the product. Thirdly, because of its high capacity, a TFDF filter is more than 10 times smaller than a depth filter, and therefore it has very low holdup volume. Together, they more effectively separate the cells away from the product. So I want to show you a graph that brings out the advantages of TFDF filtration in a bit more detail. The x-axis of the plot is time, and then we have 2 different y-axes. The first is the pressure, and the second is the flux. TFDF typically delivers flux rates up to 10 times greater than depth filtration. In this example, flux starts at 750 LMH and remains stable at that value over the course of the entire run. On the left axis, let's look at TMP. In contrast to a dead-end depth filter, TFDF directs the great majority of the cells away from the filter rather than into the filter. The product and cells therefore do not compete for access to the same channels, and the system becomes very robust to fouling. TMP for the run starts at a very low value of 0.5 psi and increases 0.25 psi at the end. So it's really low. This is essentially no fouling of the filter. The last point here is time. With a flux of 750 LMH, the operation can be completed very quickly. In this example, 5 liters was clarified using a 50 square centimeter filter in less than 1 hour. To put that in perspective, the filter for 5 liters is about the area of the palm of your hand. The combined depth filter with tangential flow, the cells going back to reactor, not getting stuck, enables it to process very quickly and be that compact. Let's take it one step further. TFDF returns the cells to the bioreactor and does not foul, which begs the question, is this process gentle enough to reuse the cells? We're all in quarantine, well, still are, after 9 months, and lots of people are making sourdough bread. My wife is also on the bandwagon, and she keeps the starter in the fridge and feeds it every once in a while so she can do multiple batches of sourdough bread. So can lentivirus production be run in this high productivity kind of mode where you can get multiple batches out of the same cells? It's also referred to as batch perfusion to some. Well, the answer is yes. Using a 5-liter sub single-use bioreactor and an HIV GFP virus, a first harvest was executed at 750 LMH, delivering a yield greater than 90%. The fresh media was added to the bioreactor, cultivation was continued, and then a second harvest was executed once again at 750 LMH. Without optimization, the yield for the second harvest was 85%. So there's a little math here, so let's add up the numbers. If you combine the first and second harvest, then you obtain a harvest yield of 170%. If we then divide this by 75%, which was the yield from the depth filter, we see that using TFDF generates nearly 2.5 times more material. Granted, there are some additional steps are taken, such as adding more media, but the value of 2.5x should far outweigh that effort. From a technology level, that the low shear rate of TFDF preserves the integrity of the HEC 293 cells, even when operation at high flux, enabling altogether new lentivirus bioreactor applications. So earlier, I showed you a graph before of one harvest of pressures and flux versus time. So I want to show you the exact same graph here, but for the two harvests that we just described in the last slide. During harvest 1 at 700 LMH, TMP increases less than 1 PSI, again, essentially no fouling. Over the course of the second run, it rises a bit, but still only 2 PSI. Therefore, both harvests are completed with the same filter with a total of less than 3 PSI increase. So it's important for me to mention that the flow path that surrounds the filter is closed and gamma irradiated, which is a key enabler of these multiple harvests. We can essentially make the connection of the TFDF to the bioreactor and then process over extended time periods with negligible contamination or bioburden risk. So all the runs that we've looked at thus far were at the 5-liter scale. So are we able to show the same performance at larger scale? So we scaled the 5-liter tenfold up to a 50-liter scale reactor and found that the yield, the process time, and the filter fouling were very similar to the 5-liter scale. So yield, again, was greater than 90%, shown here in the bar graph. Flux was constant at over 750 LMH. And TMP ended below 1 PSI, and the operation was wrapped up in less than an hour. So the TFDF filter sizes are available at larger sizes, such that volumes of 200 liters and 500 liters, even up to 2,000 liters are feasible with the TFDF technology. So yes, it's scalable, which is also really will help you in your increase in your productivity. So the bigger we go in scale, the more challenging the operation becomes. Almost every process can be managed at small scale, not too difficult. It's at larger scales that where the rubber really meets the road and where TFDF really shines. Therefore, I just want to walk through what TFDF looks like if we were to scale another tenfold from 50 liters to 500 liters. We scale the TFDF process by adding more filtered tubes to the same enclosure. This means that the simplicity of TFDF carries from the 1 liter scale to the 50 liter scale to the 500 liter scale and beyond if needed. At each of these scales, you only have one device to unbox, install, and break down. Assembly and disassembly are completed in 20 minutes with only 3 connections using genderless aseptic connectors. All the pressure sensors, the magnetic pump head are already built into the flow path. In terms of space, the entire footprint of your skid and your filter is only about 10 square feet. This is because the filter mounts onto the skid with the pumps. It's really, really, really simple. And that simplicity scales. Also, there's no need to organize 20 parts during setup. There's no Wi-Fi flush. The filter ships dry, so it saves tons of very expensive water. There's no product flush at the end. There's no product mess spilling out during the breakdown. I've seen where you take apart, filters and you just have this mess of virus spilling out everywhere. there's no hardware dedicated to just holding the filters. That core concept of combined depth and tangential flow filtration create capacity and volumetric throughput that delivers more product much faster and in a much smaller footprint than classic depth filtration. Who couldn't use more product, extra time in their day, and extra room in the lab? It's obvious, right? So I'd love to dig deeper and go deeper into how the technology works and its other applications. But I'm going to have to wrap it up with this slide that simply describes the different components that work together to create the complete solution. It starts with the tubular depth filter in the blue there on the left. The first depth filter, let me emphasize, the first depth filter to act in tangential flow mode. We then take the TFDF filter, put it into a flow path that includes the filter, obviously. It has filter, it has tubing, clamps, pressure sensors, aseptic connectors, and the single-use pump head. So all of these put together, gamma rated, ready to use. The beauty is that at the small scale, you only have one device to unbox, install, and break down. And at large scale, you still only have one device to unbox, install, it's fast, just 3 connectors, putting it onto the skid, and to break down. Again, the ProConnex flow paths are all closed, gamma radiated, and they come as both stocked off the shelf and configurable models. You can configure the flow path, the tubing sizes, the aseptic connectors. The final component to the solution is the system, which includes automated process control logic specifically engineered for TFDF. TFDF systems, like the flow paths, scale from the lab to the pilot and then to large process scale. So just a quick acknowledgement to the several contributing authors to the Cell and Gene Therapy Insights publication. Just want to say thanks to Thomas Williams. These are from Oxford Biomedica. So Thomas Williams, Oliver Goodyear, Lee Davies, Carol Nevelman, Kyriakos Mitrofanous, James Miskin. They helped with in the publication of this paper. Finally, thank you for taking a few minutes to join us for this gene therapy showcase of Repligen technology. If you're interested in learning more about TFDF or exploring new applications, reach out to our, the salesperson or visit repligen.com/TFDF and you can get more information on our website. Thank you very much.
Thank you, Michael. Next, I'd like to welcome Jason Rodriguez, Senior Manufacturing Engineer at Genopis. Jason will discuss his experience with determining the right filtration technology, hollow fiber filters or flat sheet cassettes for his UFDF steps.
Hi everyone, my name is Jason Rodriguez and I work for Genopis Incorporated. Today I will talk about the design and optimization of platform plasmid DNA tangential flow filtration processes from cell harvest to final formulation. Before I begin, I wanted to give special thanks to Repligen and my colleagues at Genopis for making these experiments possible and putting all the data together. Let's start with some information about my company, Genopis. Genopis was first established in August 2018 through a joint venture partnership by Helixsmith, formerly Biomed, and Medivate Partners. We specialize in plasmid DNA-based technology and provide contract development and manufacturing organization opportunities. Here at Genopis, our facility has a fully functional cGMP manufacturing suite with capabilities well suited for microbial-based products and expandable for future mammalian-based products. We actually recently completed construction of our new clean room that will house a small volume manufacturing train up to 30 liters of fermentation for microbial applications. So let's start with our current process and challenges. So currently we're looking at 3 steps that we want to optimize and some challenges that we're facing. So the first one is upstream process for our cell harvest clarification. So currently what we do is we use centrifugation to separate our microbial cells from the fermentation broth. So the issues with centrifugation are that it's difficult to scale up and expensive. And what we notice is difficult to find a single-use option for microbial-based applications. So what we're noticing is that for single-use options, they don't reach the necessary separation forces for microbial-based separations. So that's why we're kind of looking for an alternative for centrifugation, especially when we start scaling up and scaling down. And it provides a single-use option for our clients. So this is the reason why we turn to microfiltration as an alternative. For downstream, we have 2 UFDF steps that are not optimal. So for one of the UFDF steps, we use a hollow fiber as well as the second one. So what we would like to do is define a process for both hollow fibers and flat sheet cassettes to provide processing flexibility for our clients. So in addition, not only are our parameters not optimized, we want to be able to reduce our processing time and maximize our yield and flux. So let's start with some basic information about our product. So upstream, we use, as I mentioned before, we use microbial cells, and specifically we use intact E. coli cells. So what we're harvesting are the intact E. coli cells using centrifugation. Downstream, our target product is supercoiled plasmid DNA. So depending on which UFDF step you're on, it'll be in a different matrix. For the UFDF 1 step, it is post-defiltration after we lyse the cells. For our UFDF 2 step, it is after our second chromatography step. So it's sitting in the chromatography step to eluate. So what are we trying to achieve with these experiments? As I mentioned before, for upstream cell concentration, we want to replace centrifugation with an alternative that's feasible and that will reduce our processing time while still recovering our product. In addition, we want to create a more automated process and provide a single-use option for our clients. For downstream, we want to be able to provide processing flexibility by defining a process for both hollow fibers and flat sheet cassettes. So let's start with what our experiments entail. So each study had 2 parts. The first part was the optimization study, where we determined the critical flux for microfiltration and the optimal TMP for UFDF. The second part consisted of a full process run, which included concentration and diafiltration that was either controlled by constant permeate flux for microfiltration or constant TMP for UFDF. So let's start with our cell concentration. As I mentioned before, our main objective was to prove feasibility of microfiltration as an alternative to centrifugation. So the way we designed these experiments was we asked ourselves a few questions. So some examples include, what's a reasonable processing time? Can we reach our target concentration factor? How can we minimize our buffer usage? What is a reasonable final volume of product to store? For example, if we have 30 liters of fermentation broth, is 5 liters a reasonable volume? So the difference between microfiltration and centrifugation is centrifugation, we would typically get what we call cell paste, whereas using microfiltration, we would get more of a slurry. So it's a little bit easier to store the cell paste, but because the volume is the amount is much lower. But as I mentioned before, it's not exactly a single-use system, and microfiltration, the advantage that it also offers is that it kind of maintains more of a closed system. You don't have to be transferring out the product in and out of bags or from a container or a vessel to a bag. So for the optimization study, we decided to go with a spectrum hollow fiber with molecular weight cutoff of 500 kD to determine the critical flux. For the small scale study, we decided to target a 6x concentration followed by a 2 divolume wash. So as you can see on the slide here, we did 2 small scale studies for our microfiltration. So the first one, there was a little bit of issues, even though we were able to achieve our target. which led us into the reason why we decided to do a follow-up study to further optimize the microfiltration process. So let's start with the optimization study. How do you determine the critical flux for microfiltration applications? It's very simple. So usually what you do is you will incrementally increase your permeate flux, and you kind of watch your TMP over time. What you're looking for is for your TMP to start to increase and become less stable. So as you can see from the graph, this highlighted column, at 40 LMH permeate flux, you'll see your TMP start to become a little bit less stable and start to increase. And if you go even higher permeate fluxes at 45 LMH or higher, you'll see your TMP start to increase a lot faster and become less and less stable. So based on this data, we determined that our critical flux to be 40 LMH. And based on that, typically what you want to do for microfiltration applications is operate at a permeate flux that is lower than your critical flux to ensure you don't foul when you concentrate. And usually values range between 50 to 75% of your critical flux. We decided to go with 75%, so our small scale study would be performed at a permeate flux of 30 LMH. So how did our small scale study run go? As I mentioned before, we targeted a 6-fold reduction, but we actually achieved a 6.7-fold reduction. The only issue that we ran into was that diafiltration started a lot earlier than we had hoped. We originally wanted the diafiltration to start at 6x concentration, but we had to start it at 1.9x because we saw a significant increase in pressure, which was indicative of membrane fouling. So something to note that may have led to the reason why the membrane fouled so early was because if you look from the graph, we initially started our concentration at a permeate flux that was higher than 30 LMH. The reason for that was our permeate pump calibration was slightly off. So what we did to offset that was we slowly decreased our permeate pump until we achieved a 30 LMH permeate flux. So the fact that we started higher could have possibly fouled the membrane earlier than we would have liked, and that kind of is what led us into starting diafiltration earlier. So given that, the fact that we started diafiltration at 1.9x kind of threw a curveball in the sense that we would have to use more buffer if we were to scale up to, let's say, 30 liters or 300 liters. It would be a lot more buffer consumption, and we want to avoid that. This is the reason why we had hoped to diafilter at 6x. So this led us into our second run. So for the second small scale study, we actually decided to change the hollow fiber length. The reason was because if we were to scale up to 300 liters or 30 liters, the only available hollow fiber through Repligen at that size has a longer hollow fiber length. So if we were to use the same length, this would ensure direct scale up and see how an increase in hollow fiber length affects the process. So what happened exactly? So we were able to start diafiltration at a much later stage than 1.9x. We started diafiltration at 3.5x instead of 1.9. And we were still able to achieve our target concentration factor of 6x. Our initial flux was 25 LMH, and we ended up decreasing it a little bit to 20 LMH, which gave a total processing time of a little bit less than 4.5 hours. In addition, just as we did for the first run, we didn't see any product loss. So these two studies kind of confirm that we can use microfiltration as an alternative to centrifugation. Let's move on to our downstream portion. As I mentioned before, the material at this stage is after we had lysed the cells and we clarifying it using defiltration. So this is our clarified lysate material. So just as before for microfiltration, this study consisted of two parts, an optimization study and a small scale run. In the optimization studies, we tested 3 memories with molecular weight cutoff of 100 kD. We tested a Spectrum hollow fiber, the Tangenix high stream flat sheet cassette, and the Tangenix pro stream flat sheet cassette. So the difference between the high stream and the pro stream is more to do with their matrix. They're both based on modified polyether sulfone. However, the high stream has a hydrophilic modifier that makes it more comparable to cellulose-based membranes. The pro stream has a different modifier that gives it more of a neutral charge for low protein binding. So for each membrane, we determine the optimal TMP that will maximize our permeate flux at a given cross-flow rate. For the small-scale study, we wanted to achieve a 9x concentration prior to starting the 5-diafiltration wash. However, we were a little bit flexible. If we were to start diafiltration earlier, it would still be okay depending on what concentration factor it would be. So let's get into the small-scale study for both the hollow fiber, or the optimization study for the hollow fibers as well as the flat sheet cassettes. So the optimization study for UFDF is similar to that of microfiltration, but instead of determining the critical flux, we determine the optimal TMP that maximizes permeate flux. So basically how you do that is you increase the TMP gradually, maybe by 5 PSI increments until the permeate flux starts to plateau. And we did this for 3 different cross-flow rates for the flat sheets and 3 different shear rates for the hollow fiber. So what are you looking for to determine what is your optimal shear rate or your optimal cross-flow rate? So what we're looking for is a cross-flow rate or shear rate that provides a reasonable sweeping action to minimize fouling on your membrane, while at the same time maintaining a reasonable operating pressure and permeate fluxes. So if you can see from the graphs, you ideally would like to get your permeate flux at the knee of the curve, because you will get the maximum permeate flux at a given TMP. Because if you go a higher TMP, your permeate flux is not increasing, it's more plateauing out, so it doesn't matter. Increasing TMP would not increase your permeate flux to a certain point. So based on this data, we determined that the optimal TMP for the hollow fiber was 5 PSI at a shear rate of 6,000 inverse seconds. Now the Tangenics ProStream and HighStream flat sheet cassettes kind of perform similarly, but we only wanted to use one of them to proceed for our small scale study. So the HighStream had slightly higher permeate fluxes, so we decided to go with the HighStream. So we determined that a cross-flow flux of 5 and a TMP of 15 PSI was optimal for the Tangenics HighStream cassette. So how did the small scale study go? Well, for the hollow fiber, we were able to successfully concentrate to 9x and then perform a 5-die volume buffer exchange without any issues. The total processing time was a little less than 2.5 hours, and which is significantly less than what we currently do. However, for the Tangenics HighStream cassette, we kind of ran into an issue around a concentration factor of 6.5x. We started to notice a significant increase in pressure, as well as a significant decrease in our permeate pressure. So what we did was we decided to start diafiltration at this point to help recover any fouling that may have occurred. However, with less than 1 die volume exchange, we noticed that the feed pressure still started to increase and our permeate pressure was decreasing even faster. So this is the reason why we decided to stop the run and just go with the hollow fiber. So this doesn't mean that the cassette doesn't work. It just means that we would need to do some more follow-up studies to determine when would be the best point to start diafiltration to help minimize this fouling. The good thing is for both the hollow fiber and the Tangenics HighStream, we did see no product breakthrough in the permeate stream. Most of what we see in the permeate is usually RNA, because at this stage in the process, you have lots and lots of RNA. So our total recovery for our hollow fiber was about 60%, which may seem low, but that is typical for what we see at this step, because there's so much RNA, genomic, and we can't, at this stage, we don't have the analytical tools to quantify RNA versus genomic and versus plasmid. So let's get into our UFDF2 step. So this material is coming off of our second chromatography step. So just like before, we did an optimization study and a small scale study. For the optimization study, we used a 30 kilodalton molecular weight cutoff membrane, and we tested both a Spectrum hollow fiber and a Tangenics HighStream. We decided not to test the ProStream because, as we saw for UFDF1, the HighStream showed a little bit higher flux. So that's what we wanted to go with. For the small scale study, we wanted to achieve a final overall concentration factor of 8x or higher. Diafiltration, we were kind of open to when it would start, ideally if we can get it at 8x, but anytime before, so long as the buffer usage was reasonable, it should be okay. So the optimization study for UFTF2, it was done in a very similar manner as UFTF1, where you test 3 different shear rates for the hollow fiber and 3 different crossflow rates, and you look for the knee of the curve to kind of maximize your permeate flux. So what we found for the hollow fiber was that the shear rate of 6,000 inverse seconds and a TMP of 7.5 PSI was optimal. For the Tangenics high stream flat sheet cassette, we found that a crossflow flux of 5 and a TMP of 12 PSI was optimal. So what happened when we use these parameters for our small scale study? So on the top, you'll see the hollow fiber data. We were able, for both actually, we were able to successfully concentrate to 8x with the diafiltration occurring at 6x concentration factor. We were able to successfully do the 9 die volumes exchange, and the processing time for both the hollow fiber and the Tangenics flat sheet cassette were less than 1 hour. The average permeate flux for the hollow fiber was 41 LMH, and the average permeate flux for the Tangenic was 51 LMH. Now, although both were able to complete successfully, we did notice something interesting in the analytical results of the permeate stream. So from the gel on the right, there seemed to be some product breakthrough in the permeate stream for the Tangenics high stream cassette. However, if you look closely, the migration is not exactly where the supercoiled plasmid DNA should be. It's kind of in between the open circular and the supercoiled. So what our analytical team found or thought was this may or may not be actual product breakthrough. What it could be is interference from the sample matrix. So what that means is basically sometimes when you have high salt in your sample, it can interfere with the migration of your sample. And given the nature of our permeate stream, there is definitely high salt in there. So we thought that there was no product loss. So to confirm that, we decided to run HPLC. Now, so the top graph is of the hollow fiber. So you see there's no fourth bar graph showing the permeate stream. So the first one is your initial sample. The second is our retentate sample, and the third is from our recovery flush. So basically what that means is you concentrate the product, you collect it, and then you add some buffer and then recover that. And those all showed that we had a plasmid and all the conformers in there. Now for the Tangenics, you'll see there's a fourth column to each of them. So this means this fourth column is the permeate stream. So for the Tangenics, you definitely see some supercoiled in there and some of the other conformers as well. So this resulted in a lower yield for the Tangenics high stream flat sheet cassette. So for the hollow fiber, our final product recovery was 84% versus our Tangenics high stream, which was only 65%. And this is likely due because we saw product breakthrough. So we're not entirely sure why there was product breakthrough. There was it could be any number of things. It could be just the nature of our product, or it could have been we're operating at higher pressures than we would have liked and possibly squeeze the product through. There's still further investigation that needs to be done to figure out why the Tangenics cassette shows some breakthrough, because when we did our optimization study, we did test the concentration of our permeate stream as well to determine if there was any breakthrough, and we did not see anything at that stage or at that point. So this kind of threw a curveball at us in trying to figure out as to why there was product breakthrough in our Tangenics flat sheet cassette. So we will likely have to do some more follow-up studies if we want to proceed with developing a method using flat sheet cassettes. So what did all this data tell us? Essentially, what it told us is that hollow fibers seem to be recommended for the entire process, whether we're doing microfiltration or the UFDF steps, especially for processing plasmid DNA. If we were to use cassettes, we would have to do a lot further investigation and optimization to ensure we reach our targets and there's no product loss. So in the first table below, you'll see some scale-up scenarios we did for microfiltration for a 30-liter scale and 300-liter scale. And by that, I'm referring to fermentation. So all two scenarios show the hollow fiber with the longer fiber length, just as we did for our second small scale study. The table at the bottom shows scale up information for our 30-liter scale for all 3 unit operations. And this aligns almost perfectly with the new capabilities that our new clean room has. So let's go over all the results that we got from these studies. So for cell concentration, we were able to achieve our target concentration factor of 6x, and we were still able to do a 2-die volume exchange with no product loss while minimizing our buffer usage. In addition, we were able to prove feasibility of microfiltration as an alternative to centrifugation. Our processing time was a little bit less than 4.5 hours, which is still reasonable and typical of filtration operations. For our UFDF1 step, the Spectrum hollow fiber was more successful in completing our 9x concentration, followed by a 5-die volume wash. The yield was a little bit low, but that's expected for this stage in the process, as what we can only measure is total nucleic acid, and we cannot resolve the quantities of all of the RNA genomic versus plasmid. So the TangenX high stream cassette for this particular process did not work or did not complete what we wanted it to do. It did not achieve the 9x, and we were unable to perform the diafiltration due to significant pressure increases. So in order to mitigate that, we would have to do more further optimization, follow-up studies to figure out what is the best way to operate the TangenX high stream flat sheet cassette. For the UFDF2 step, like the UFDF1, the hollow fiber was more successful in completing the concentration diafiltration because we achieved a higher yield. Although the TangenX high stream cassette was able to concentrate and diafilter as we wanted to, there was product breakthrough, and that's the reason why we need to further investigate why that was. So based on this data, it is recommended that we use hollow fibers for all 3 applications. I would like to thank my colleagues from Genopus for their support during these studies, as well as Repligen's field applications team. I wanted to give a special thanks to Monica Amin from Repligen, as she was a key member in executing all these experiments and supporting the data analysis. Thank you everyone for your time.
Thank you, Jason, for this talk illustrating the importance of identifying the technologies appropriately suited for the different steps in your processes. Next, Dr. Tanya Buch, Technical Director at the Biofactory Competence Center, will discuss about process monitoring and control of bacterial fermentation processes, which is key for consistent quality of plasmid DNA used in gene therapy.
Welcome everybody to this technical seminar from the Biofactory Competence Center in partnership with T Technology on the topic monitoring microbial growth using variable path length spectroscopy. My name is Tanya Buch and I'm the technical director of the BCC in Switzerland. To start with, I would like to say a couple words to myself. I'm holding a master's degree in bioprocess system engineering from the University and Max Planck Institution in Magdeburg in east of Germany. After gaining some industrial experience in Switzerland, I continued my studies in Ireland at the Dublin City University, where I completed my PhD in bioprocess engineering on an industrial project in cooperation with the company MSD. Throughout my time in Ireland, I was involved in several industrial projects, as well as a trainer at the Training and Research Centre NIBRD in the south of Dublin. Here I developed and delivered applied training courses to the entire biopharma sectors in Ireland. In 2015, I decided to follow my colleague Ian Marison back to Switzerland and helped him to develop and set up the Biofactory Competence Center in Fribourg. And since 2016, we are operational in providing training and research services to the biopharma industry. This brings me to my second introduction here. What actually is the BCC? So the BCC is a non-for-profit organization supported by the cantonal government in Fribourg with the aim to support the pharma and biopharma market and industry in Switzerland and also abroad. For this, we have 3 main activities. Our main activity is the provision of training courses which we give to the biopharma sectors. For this, we have constructed a GMP-like facility in our plant in Fribourg. These are not meant to operate as a GMP factory, but we try to simulate them as much as possible to a GMP process. In these labs, we can provide training courses on all different aspects of a biopharma process, starting from seed train development over USP processes, down to cell separation and all the way to product purification on filtration or chromatographic technologies. For this, we also have two different USP suites, one for mammalian processes and the other one to cover microbial processes. Our training can be adapted to all different levels, for example, for operators or even cross-training courses, where we train people on GMP principles, how to behave in clean rooms, or how to work in an aseptic manner. We also give more expert training courses, for example, on continuous processing in fermentation or cell culture processes, but also on very technical steps, for example, in continuous chromatography. We give these training courses in 3 different languages. Since Fribourg is right at the language barrier between French and German, we provide these courses in either English, French, or German. The duration of these training courses can also differ between 1 day and up to 6 weeks, completely what the client requires for the training courses. Our second activity of the BCC is the provision of technical service to the biopharma industry. Here we try to support the industry in process development or process optimization of key steps and try to find solutions how they can overcome these challenges in their processes. Another part of the technical service is also the development or optimization of analytical methods, which become more and more important in monitoring and control of your processes. We usually have an internal joke in our company which says, just ask. So meaning, if you have a problem in your process, just approach us and either we can help you or we try to find a solution for you with one of our collaborators. Our last and third activity is the design of modular and flexible facilities, which can be used either for training, research, or also for production. And here we finally kicked off this process with a contract that we signed end of last year, where a local pharma company in Fribourg decided to extend their production facility and wanted to build a new plant with over 1,000 square meters. And they decided to go ahead with our modular design, which should be finished and operational by mid of next year. You may ask yourself how the collaboration between the BCC and C Technology originated. In 2018, one of our clients that we gave training courses for approached us because they heard about the variable path length technology and they wanted to have this technology implemented in their training programs that we gave to them. Since at that time, we didn't have such technology available in our labs, we approached C Technology and asked them if they would be interested to collaborate with us and to support us with one of their systems so that we can implement it in our training programs, but also offer it as services to our clients. And C Technology liked our ideas and they gave us first the Flow VPE system, because that time we had a very high focus and demand on continuous DSP. And the Flow VPE system is a brilliant tool in order to include it as a monitoring and control tool in protein concentration throughout your continuous purification process. And in our training courses, we implemented it, for example, after a single pass TFF system in order to follow the protein concentration in real time and to see, do we really have a consistent protein concentration coming off this filtration cassette, or does it vary over time? We also implemented the flow prior to a continuous chromatography device, here not as a monitoring system, but more as a control device in order to control the protein load onto the columns and to avoid overloading your columns throughout the continuous process. Since this collaboration worked really well, C Technology decided to also provide us with their benchtop device, the Solo VPE system. And having this benchtop device now in our labs, we could even go one step further and we could think, what alternative application can we use the variable path length technology for? And one of the first technologies that came to mind was measuring the cell growth of microbial processes. As you probably know, usually we are using a standard spectrophotometer and follow the cell growth with an absorbance of 600 nanometers for E. coli cells, but you always have to dilute your samples already at the very early stage of the process. So we thought, why not try to adapt this variable path length technology and eliminate this dilution step from our process and therefore become a higher accuracy in our measurement and a new tool in order to monitor and ideally also control our microbial processes. You may think now, what does microbial fermentation have to do with gene therapy and viral vector production? But as you know, you always require a certain amount of plasmids in order to deliver the transgene or also the plasmids that encode for our viral particles into our mammalian cells or insect cells for the viral vector manufacturing process. And these plasmids are actually produced with a microbial process, in most of the cases with an E. coli fermentation. And since the demand and the interest in viral vector therapies is increasing over the last years extremely, there's also a really high need for large-scale production of plasmids. And therefore, we require efficient E. coli fermentations, and the variable path length spectroscopy technology can nicely be adapted and implemented in order to monitor, but ideally also control the cell growth of our E. coli cells throughout the USP part of the plasmid production part. And this is why we cover this here today. So just to recall before we go into all these details, why monitoring and control is so essential. As we all know, cell concentrations and cell growth are one of the important critical process parameters which have a significant impact on the critical quality attributes of our products. And regulatory agencies such as the FDA or also the EMA expect more and more good process understanding and process control, which we can only provide if we have good analytical methods. So to give you one example, when we talk about fermentation processes, we always usually separate them in two different phases. The first phase being the cell growth part and the second phase being the product production part. And the different phases are divided with an induction time point, which can be dependent on a temperature shift or also on a substrate shift in order to change the metabolism. But in most cases, they are dependent on the cell concentration present in our bioreactor at this given time point. So if we look at our lower right-hand image here in this graph, and we, for example, identified that the ideal time point for the induction is at an OD of 0.5, we see it's right in the middle between two sampling time points. So when we took a sample after 90 minutes, we have only an OD of 0.4. So we have to make the decision now, do we induce already now in order to start the production of our product, or do we wait until the next time point? If we start the induction too early, it means we don't have enough cells present, and lower cell number means we are also consequently producing less product. However, if we wait 30 minutes until we take the next sample and then we have an OD of 0.6, we see here already we are out of the exponential phase, going into steady state, which means the metabolism is slowing down. Some of the cells may go in apoptosis, and this can lead either to inefficient product formation, or some product may not have the same quality as we need it to have. So therefore, if we have reliable and good accurate methods, can definitely be improvement in monitoring and controlling our microbial growth process. So let's look at the current technologies that we have available in order to monitor the microbial growth. The most method, it's probably the offline method using a standard spectrophotometer and measuring the optical density of our cell solution at different wavelengths depending on the cell line. For E. coli, for example, at 600 nanometers. However, the drawback here is that this linear relationship from a standard spectrophotometer between the optical density and our cell concentration is only up to an OD of 0.5, meaning all our fermentation samples that exceed the OD of 0.5 need to be diluted, which we can also see in this little graph on the lower and left side corner, where we see in blue our optical densities without making any dilutions, even though the cutoff of 0.5 is reached. In green, we see the example where dilutions have been made prior to the optical density measurement. And as we can see, after the cutoff of 0.5, we see already a clear distance between the measurement without and with the dilution. And this is getting more and more significant as long as the process lasts. So this clearly shows after the cutoff of 0.5, we just have to make dilutions for this step. But let's look what we have for online method nowadays available. Currently, the push goes into photometric sensors, for example, the optical probes from Mettler Toledo, which become more and more interest in industry and also are started to be applied by a lot of different industrial companies because they have good accuracy and they also are robust over a certain time of the fermentation process. However, also these optical probes have some drawbacks. First of all, they have their limitations with high cell density cultures, and they have the tendency of material buildup around the sensor, especially on the optical lens at the bottom. In addition, if you require a lot of oxygen and you need a high aeration rate, these bubbles can also interfere with our measurement. But one of the biggest drawbacks of these optical probes are that we are not measuring one specific point, but we are always measuring a spectrum at each given time point when we have to measure our cell density. And if you measure a spectrum, you need to do chemometrics models in order to convert the spectrum into one real giving data time point, which can be equivalent to the optical density value. And these can be time consuming and also needs a certain expertise in order to develop these models. So we see there is definitely room for an alternative method and also for an improved method as offline, but also for online methods in order to follow the microbial growth in our bioreactors. So let's have a quick look how the variable path length technology works. You probably all know the principle of a standard spectrophotometer, which follows the Baird-Lambert law, where we say our absorbance equals the extinction coefficient times the concentration of our solution times the path length. Path length meaning the distance of the light that it has to travel through in order to measure the absorbance. The path length in a standard spectrophotometer is always set to 1 centimeter, due to the shape of our cuvettes, but also due to all the standard spectrophotometers, which allow only a 1 centimeter path length in the measurement. So meaning with the extinction coefficient being a constant, the absorbance equals the concentration. A nice linear relation, and this would be great. However, the problem here again is that this linear relation is only linear up to an absorbance value of 1.5 or maybe 2, depending on what we are measuring. So as soon as we have a high concentration of our product that we want to measure, we have to dilute again our sample to reduce the absorbance below 1.5 to work in our linear range, and that the Beer-Lambert law is actually valid. So SEE Technology here was very smart. They thought, what other opportunity do I have in order to reduce my absorbance value below 1.5, but without the need of diluting my sample? And of course, the only parameter left in my equation is the path length. So meaning if we reduce the path length a lot when we have a high concentration, we can reduce our absorbance below this famous cutoff at 1.5. So we are in the linear working range, but we didn't need to dilute our sample. This is also the machine now that we can see here on the upper right-hand side. We have a standard spectrophotometer, which is working here as a light source. And then we have the Solo VPE system on the top, where we see the light source is connected with the fiber optic cable to the top of the system and goes all the way down to the nozzle. And at this little nozzle, we can implement a fibrate. And the fibrate afterwards can go down into the liquid and is the extension for the light source. Our cuvettes are placed here on this little plate holder, and our detector is now on the bottom. So if we zoom in for our cuvettes, what we see down here, we have the cuvette with the process sample, the detector would be at the bottom, and our famous path length is defined from the bottom where the detector is all the way up to the end of our fibrate, which is the extension of the light source. And this fibrate can be moved up and down in very specific step paths so that we can adapt the path length according to the concentration so that our absorbance is always below the cutoff of 1.5. Question now may be, how do we know what path lengths to choose if we don't know the concentration? Very good question. And the answer here is what the Solo VPE system is doing at the beginning. It scans through our unknown process sample all the different path lengths from the bottom to the top in order to find the path lengths where we have an absorbance value of 1.0. Because we know if we work around an absorbance of 1.0, we are definitely in the linear range of our Beer-Lambert law. So we see this here on the right-hand side. The starting path length always starts up an absorbance of 1.0 usually. But instead of just having a one-point measurement, we want to improve our precision of this measurement, and we are actually measuring 10 data points instead of just 1. So we are starting with a path length at 1.0, but then we measure 9 further ones with a stepwise path length depending on the starting path length. And also the step path length is dependent on our protein concentration. So meaning if we have a high protein or product concentration in our solution, we have, of course, a very narrow starting path length in order to have the absorbance down at 1.0, but the step path length is actually very narrow as well, so around 0.05 millimeters. However, if we have a low concentration present in our product or protein in the solution, the starting path length is quite high up, probably close to 1 centimeter, but the step path length is now at 0.15 millimeters. And then what the system is doing, we are plotting the absorbance over the path length, and the relation should definitely be linear, defined by the Beer-Lambert law. And the slope out of this, which we also often call the slope absorbance, should have an R-square of at least 0.999 in order to give us confidence that this measurement works. And the slope absorbance can then be recalculated to determine the concentration value. This is how the Solo VPE system works under their default setting, and which works really well in order to determine unknown protein solutions. So we thought, why don't we try exactly the same process steps for our cell growth of our E. coli cultures? And unfortunately, with these settings, it didn't work really well. because we had quite a lot of variation between our absorbance and the path lengths, and the R-square was sometimes below 0.9, and this didn't give us enough confidence that it worked. So we actually went a step back and we thought, what are we actually measuring here? So if you have a protein in solution, the protein is soluble, and you measure the absorbance, which is defined as the ability of a medium to absorb the light. However, when we now look at our cells in our medium, these are more particles, and we are measuring the optical density, which is defined as how much light passing through a medium, which is kind of the opposite a little bit. Plus, if you have a lot of particles in the different layers, the chances of light getting through get slimmer and slimmer. So we thought, okay, why don't we just fix our starting path length? So instead of to having it variable and always starting at an absorbance of 1.0, we decided to fix it to 3 millimeters. And also the step path lengths now, we thought if we do these steps a little bit wider, we are measuring our linear range over a bigger distance, which should improve our linearity. And this is what we did. So we fixed also the step path lengths to 0.15, which is at the end the complete opposite what we did with the protein concentration measurement. But we also measure the difference when we measure the optical density. And we kept our 10 measuring points that we want to measure. So we used these new set points of our method and tried it out in an experimental fermentation with E. coli cells. So what we did, we cultivated E. coli cells in shake flask, 250 milliliter volume, total volume, standard YPG medium at 37 degrees, 150 RPM. And we took samples every 30 minutes and measured those samples with a standard spectrophotometer for the optical density values, but also with the solar VPE system for the slope absorbance value. And these were the results that we received. So on the left-hand side first, the results from a standard spectrophotometer. We measured optical density, and of course, after this famous cutoff of 0.5 was reached, we had to dilute our samples in order to get this nice growth curve of our E. coli cells as we would expect them. Of course, we also had to include here the blank correction because YPG medium does absorb the light at 600 nanometers. So you have to make the correction in order to avoid interference of the medium with the measurement. We took the same samples and analyzed them with the Solo VPE system for the slope absorbance, what we can see now on the right. And here, there was no need for diluting the samples, and we also didn't include any blank correction due to the YPG medium. And when we compare the growth curve and the trends that we can see between the optical density measurement and the slope absorbance measurement, they look very alike. So this gave us confidence that there definitely is a potential there to use the variable slope spectroscopy to measure the cell growth or to follow the cell growth throughout a microbial fermentation. The next steps that we wanted to know is, can we make a correlation between the optical density measurement and our slope absorbance values? So what we did, we took the remaining samples we had left over from our experiment. We concentrated the cells a little bit because beforehand in a shake flask, the maximum OD was only 3, and we wanted to get a little bit higher cell densities. So we concentrated them up to an OD of 25, made several dilutions of those, measured the different samples for optical density with a standard spectrophotometer, but also with the Solo VPE for the slope absorbance, and we plotted these two different values over each other. And as we can see here, we identified a nice linear correlation between the two parameters with an R-square of 0.992 and a correlation factor of 94.5. When we looked at the variability of these measurements, we saw that measuring the slope absorbance of one sample several times resulted in a variability of less than 2%. Using our correlation factor to recalculate it into an optical density value increased the variability a little bit, but was still less than 5%, which is a very good value for an analytical method. So this showed us there is a potential there. So we wanted to have a proof of concept. So we repeated our experiments in a shake flask with E. coli, the same settings than before with YPG medium at 37 degrees. And again, we took samples every 30 minutes and measured those with a standard spectrophotometer for optical density, and then also with the Solo VPE system for the slope absorbance values. And what you see now on the lower right-hand corner is in blue, the values of the standard spectrophotometer, our optical density values. Again, after the cutoff of 0.5 OD was reached, we had to dilute them, and we had to include the blank correction of the interference of our medium samples. But the growth curve was almost as expected in what we saw before, with some small deviations here around a processing time of 4 and 4.5 hours, which we probably assume that this is due to some dilutions errors that was made by the person who performed this experiment. However, we took these same samples, analyzed the slope absorbance with the Solo VPE system, and then we used our identified correlation factor in order to recalculate the slope absorbance into optical density values, and we plotted these results in the same graph and can be seen as the green dots. And as you see, they follow exactly the same trends and what we identified with the standard spectrophotometer, except our little deviation that we see here, where we assume this might be due to the dilution errors, because with the Solo VPE system, we don't need dilutions, and therefore we don't have this variability in these measurements. So these results, again, convinced us that it's definitely possible to use the variable path length spectroscopy technology in order to implement it as a monitoring tool for microbial processes, especially with E. coli cells, as we can see it here. But the overall aim was to actually not have another offline method, but the idea also is to have ideally an online method, which we cannot just use for monitoring, but ideally also as a control tool in order to run our microbial processes more efficiently. So one of our next steps that we are planning on doing is to try to implement the Flow VPE system to one of our benchtop bioreactors with a pump. We are circulating our process sample through the bioreactor with a pump to our Flow VPE system and then back to the bioreactor. And this should allow us to have an add-line tool with recirculation loop in order to monitor the cell growth in real time and use the Flow VPE data in order to control the feed that we have to start, or maybe identify the optimal induction time point in order to shift from the cell growth part to the production part of our product in our fermentation setup. With this, I'm at the end of this technical seminar, and I'm more than happy to answer any questions you may have on this topic. Thank you very much for attending, and if you have follow-up questions also later on, please feel free to contact us or C Technology directly in order to help and support you if you want to implement this method in your microbial process.
Thank you, Dr. Bock, for this wonderful presentation. And thank you all for your participation. We look forward to seeing you at our next Repligen seminar.